This tutorial will focus on a particular tool used by geneticists to amplify, that is, increase the amount of DNA available to them if they only have a very small quantity to start with. For example, from a crime scene, they may only have a very small sample, and so they can work with it on other tools like gel electrophoresis and so on, where they need larger quantities of DNA. They use PCR, or polymerase chain reaction, as a way of making many more exact copies of the original DNA. So they can turn a single strand of DNA into millions and millions of copies, exact copies, within a very short time in a machine. So let's have a look at the process quickly. It starts with having your original strand of DNA, shown here. So you've double-stranded DNA, and this DNA is initially denatured by heating to about 95 degrees Celsius. Now, denatured in this context means to separate. The heat is applied and it breaks the hydrogen bonds down the middle of the DNA and separates the two strands of the DNA. Once the strands are separated, they lower the temperature to about 60 degrees Celsius, and then they add into the mixture what are called primers. These primers can be a couple of different possibilities. Sometimes the primers are single-stranded DNA, so DNA that's been constructed artificially or synthetically by adding DNA nucleotides together just to create a single strand, so ssDNA. And these primers are very short and are designed to just anneal, is the word, that is join to the complementary bases along the original strands. The next step is to raise the temperature to about 72 degrees Celsius, and just like in DNA replication, where new nucleotides have to be added, in the PCR, new nucleotides also have to be added. And to do this, an enzyme is required. Normally, DNA polymerase is the enzyme that's used to add new nucleotides to the sequence. In PCR, often a specialized version of DNA polymerase is used, an enzyme taken from thermal bacteria, thermally aquatic bacteria, in other words, bacteria that can withstand very high temperatures. So they've evolved enzymes that do not denature at the high temperature of 72 degrees Celsius. This is often referred to as TAQ, T-A-Q. I'll just rub that out for a minute. So TAQ, TAQ polymerase, it's called. The TAQ comes from the initials of the species name of the bacteria, something like Thermo aquaticus. So a bacterium that can survive high temperatures. Anyway, it's a kind of DNA polymerase, and so TAQ polymerase is added, and then new nucleotides, individual nucleotides are thrown into the mix, and these nucleotides will then anneal, extending from the primer here, and then get added by the TAQ polymerase across the length of the DNA. So what you end up with is two new strands of double-stranded DNA, each of them identical to the original copy up here. And what happens is they repeat this cycle 25 to 35 times, and this amplifies the original piece of DNA, that is, it increases it in numbers, and it gets it up into sufficient numbers so that it can actually be used, as I said, in techniques like gel electrophoresis where it can be visualized. So it's a very powerful genetic tool.